Greetings everyone and a warm welcome to sqadia.com. Today, in our carbohydrate metabolism series, we will be talking about glycogen metabolism. Well, glucose can be interconverted into glycogen. This is the storage form of glucose in the body. Whenever there is a need of glucose to be converted into glycogen, the process is known as glycogenesis. And whenever this glycogen is need to convert back to glucose, then this process is known as glycogenolysis. We will highlight glycogenesis today, and in the next video we will be talking about glycogenolysis. So before understanding the whole mechanism of glycogenesis, first we need to know what is glycogenesis. Basically, it is the formation of glycogen from glucose molecule. If we look at the general structure of glucose here, it is a 6-carbon compound. Let's name them over here. 1, 2, 3, 4, 5, and 6-carbon compound. And on the alternate position, there is presence of hydrogen and hydroxyl groups. Now, every glucose molecule have a glycosidic bond between them. To understand that, let us take an example of a glucose molecule. You can see that one position of one glucose molecule is alternate to the fourth position of other glucose molecule. Now, hydroxyl groups are present in both same positions, so the bond, if they're going to form a bond between each other, it will be an alpha bond and it will be a glycosidic bond. It should be kept in mind that glucose always make a glycosidic bond. Now, what will happen is that water molecule will be eliminated in this reaction, and ultimately both of these glucose molecules will combine together. Now, this bond is known as alpha-1,4 glucosidase bond. One more thing that a glucose molecule can do is the formation of bond in the position number 6. Let us take one more glucose molecule. In here, the position 1 and position 6 will combine together to form an alpha-1,6 glucosidase bond. You can see the bond over here. So that's how glucose is going to make two types of bonds, which will be at position number 1 and 4, and the other one will be at position number 1 and 6. Combination, a large or vast combination of these molecules will ultimately form glycogen. So now that you've understood the bond between the glucose, let us talk about the process of glycogenesis. So here is a glucose molecule. Now this glucose molecule will enter the hepatocytes. We are discussing it inside the liver. So it will be converted into glucose 6-phosphate with the help of enzyme glucokinase. Remember that the glucokinase enzyme, whenever it's present in the muscles, it's known as hexokinase. But whenever it is present in liver, it will be called as glucokinase. So, this glucose 6-phosphate will convert itself into glucose 1-phosphate. And this will be aided by the enzyme known as phosphoglucomutase. Now, mutase enzyme has just changed the position of phosphate group from 6 to 1. In the next step, this glucose 1-phosphate will combine with uridine triphosphate. Now, uridine triphosphate is the presence of uracil with 5 ribose sugar and 3 phosphate groups. So in combination, it is known as uridine triphosphate. When it will react with the glucose 1-phosphate, what will happen is that 2 phosphate group will be removed. One will be removed from the first position of glucose, and the second one will be removed from uridine triphosphate. Now, both of these molecules will combine and form uracil diphosphate glucose, which means there is addition of uridine diphosphate into glucose. Now, After fusion of both of these compounds, this uracil diphosphate glucose is formed, and it is done under the enzyme known as UDP-glucose phosphorylase. Remember that phosphorylase enzymes are usually helpful in removing the phosphate group from a specific position. Now let us revise the process first. The glucose was converted into glucose 6-phosphate. Then glucose 6-phosphate was converted to glucose 1-phosphate. Glucose 1-phosphate reacted to uridine triphosphate and it was converted to uracil diphosphate glucose. Now, this uracil diphosphate and glucose bond will be broken, and this will be done by the enzyme known as glycogenin. This glycogenin will do two things. First, it will cleave the bond of uracil diphosphate and glucose, and secondly, it is going to bind glucose on its surface. Now you can see it over here that glycogenin has bound glucose towards itself and there is removal of uracil diphosphate molecule. Now, glycogenin will undergo autoglycosylation, through which more glucose molecules will combine together like this, and there will be removal of uracil diphosphate. This bond that is formed between these glucose molecules is alpha-1,4-glucosidase bond. The series of multiple reaction is going to occur, and this is happening because of the enzyme known as glycogen synthase. Now, when there will be plenty of this reaction going on, there will be one more enzyme acting over here, which will be called as glycogen branching enzyme. It will remove the bond of alpha-1,4-glucosidase and it will shift it towards alpha-1,6 linkage, which means that it's going to remove the bond from the 4 carbon and it's going to move this bond towards 6th carbon position. Now all these 3 enzymes, the last 3 enzymes that we discuss are glycogenin, glycogen branching enzyme and glycogen synthase. All 3 will work in synergy, and there will be formation of more chains of glucose. This will ultimately lead to the formation of glycogen, which is the molecule that we require for the process of glycogenesis. So, to summarize the whole process, glucose converted into glucose 6-phosphate, then it was converted to glucose 1-phosphate, and then the action of uridine triphosphate occurred and it was converted into uracil diphosphate glucose. Then, 3 enzymes known as glycogenin, glycogen branching enzyme and glycogen synthase enzyme acted on it, and it ultimately leads to the formation of glycogen. So this was all about the glycogenesis. In the next section, we will be discussing about the regulation of glycogenesis and glycogenolysis. So stick around with us.